Hello, and welcome to another lesson on genetics. So today I will be using jelly beans to demonstrate Mendelian genetics. And this is a lesson I developed a few years ago. Actually, when I developed it, I was trying to come up with a lesson to explain Mendelian genetics to elementary school students. But what I actually like about this lesson is that it actually could be modified for high school students as well, depending on how you implement the lesson. So jelly beans are kind of fun. They help illustrate alleles, which are the variations of genes that code for a particular trait. And what I've decided to do is I've used Mendel's classic experiment, Gregor Mendel's classic experiment of pea plants, where he used purple flowered. Yes, they're blue. I made the mistake of not buying the jelly bellies with the grape jelly beans, so I found the blueberry ones. I substituted blueberry. And white flowers. So you have purple flowers and white flowers. And what I have, in fact, is two gift bags. One that's white. I happened to find a gift bag at home that was white to represent the pea plant with the white flower, and another gift bag that's purple that represents the pea plant with the purple flower. And so this is what we call the P1 or parent 1 generation. And in this case, as some of you may know, the allele for white is recessive to the allele for purple flowers. And so the only way a pea plant can express white flowers is that both of its allele, the ones that it inherits from its parents, have to have white alleles in order for it to get a purple flower. Whereas with a purple flower, the plant can either be heterozygous, meaning having one purple allele and one white allele, or of course both alleles for purple, which is homozygous. And that's what this parent is. This parent is homozygous for purple flowers represented by the blue jelly beans. And so let's say when you cross these two flowers, right? So if you just, without looking, the whole idea is to have the student pick the jelly bean out without looking, and that's why you have to use an opaque bag. Naturally, it's going to be 100% chance that this parent will pass on an allele for white flowers. And since this one is homozygous for purple, they're naturally going to have a 100% chance of having a purple flower. And so when Mendel did this experiment, he found that all the offspring had purple flowers. And that's where he came up with the concept of dominant traits versus recessive traits, or what we call a purple phenotype, but a genotype of being heterozygous, meaning having two different alleles. And the offspring, represented by this gift bag, the Santa Claus gift bag, will therefore also have purple flowers, but it will be heterozygous. Now, if any of you remember the famous Mendel experiment, what he did is he then crossbred that next generation to see if he would get the same results. Would they all come out purple? Would it be a combination of white and purple? Or so maybe something completely different. So what he wound up doing is, through this cross-fertilization, he was able to put together what we call the Punnett Square. But part of what you want to do is you want to have the student randomly. Again, they can't see the allele. They have to randomly pick it up. So let's say you have, in this case, okay, this is purple. And you also can't, make sure they don't look. And this is white. And so in this case, this offspring is again heterozygous. But again, and this helps students understand, well, you have a 50% chance of passing on a purple allele and a 50% chance of passing on a white allele, but you can have different combinations. So if you know the famous Mendel experiment, what happened is that he noticed that there was a ratio. He noticed that about roughly 75% of the offspring had purple flowers, and 25% of the offspring had white flowers. Now, again, this is where we go into that kind of modification depending on how advanced your students are. Now, if you're teaching your students, elementary students, and you just want to help students understand the concept of inheriting traits, you can leave it at here and just say, help them understand. In fact, you could probably have them repeat it many times, a few times, so that they get the idea that, oh, we could have blue and white, we could have blue and blue, or purple and white, purple and purple, or white. But because purple is always dominant to the allele for purple is always dominant for the allele to white flowers, you're going to see about roughly 3/4 of the alleles of 3/4 of the offspring will have purple flowers, whereas only about 1/4 of them will be white flowers. And so you could actually represent this through a Punnett square. So you have, again, capital P for purple, lowercase p for white. So you have the blue and the white. And over here again, blue and white. And this is where you have the combination. So purple and purple. So in this case, you have purple and purple. So this is for more advanced students, for them to understand the concept of the Punnett square. And again, you have purple and white. And over here, you have white and purple, represented by blue jelly beans. And then finally, white and white. Whereas you know that this is going to be white, this will be purple, this will be purple, and this will also be purple, knowing that the allele for purple always masks the allele for white. And this is where Mendel developed the concept of law of segregation, where he hypothesized that that parents have a 50% chance of passing on either allele because each one of them came from their parents and they just naturally separated and were passed on. So they segregated and were passed on, and hence the law of segregation. Now, and I think what's amazing about and this is something to help your students understand, that when Mendel first developed these experiments, he had no understanding of meiosis, he had no understanding of mitosis, he had no understanding of DNA and genes and chromosomes, but he was able to use math and through ratios to understand the concept of alleles and genes. And so in many ways, we have to give him a lot of credit for what he did. So how can we modify this for high school students? Well, it's just using jelly beans, I think, is definitely much more fun. But the other way you could actually modify this for high school or even advanced middle school students is to have them, again, go back to the two combinations. So you have this one again has one purple allele, one white allele. This one has one white allele, one purple allele. So both are heterozygous. What you can actually have them do is try to repeat this as many times. Like you could actually have them do this 100 times and just constantly I've seen teachers use a coin where they use heads or tails. So one of the things I always learned actually is that there's a slight increase that will be heads because the heads is slightly heavier than the tails. It's one of the things I learned in statistics class. But this is should be completely random. Let's say you do it 100 times. You know most likely you're not going to get exactly 75 purple flowers where it's 50%. It's 50% heterozygous and 25% homozygous for purple. But you know it's going to be within that range. And so this is where you can even get even more advanced. So the other thing is, of course, not only figuring out not only figure out phenotypic ratio, in this case 3 purple to 1 white flower, 3 purple flowers, 1 white flowers, you can even do a full chi-square analysis with students. And that's the great thing I like about this lesson. It's very easy to implement. It can be modified so that even an elementary school student can understand it. But it could also be modified for a high school, a very advanced high school class as well. And it's just a lot more fun when you have jelly beans and the idea of chance, of random chance. So I hope you found this helpful. And again, if you have any questions, feel free to comment below. I'll be more than happy to answer that. And let me know how this lesson goes. Thank you. Thank you for watching. Bye.